One of the things that we'd like you to do is complete the surveys. Jennifer is handing those out. It's important to maintain our CME accreditation and to get your thoughts about the content. As you know, this conference is videotaped, and we'd like you to use the microphones in the discussion for the questions. All right. I think we'll go ahead and get started. It's a real pleasure to introduce Professor Alan Daugherty to you today. He is a major figure in the biology of atherosclerosis. Has contributed a lot to our understanding of the pathobiology of aortic aneurysms and atherosclerosis. He grew up in Liverpool, did his PhD training at the University of Bath, and then immigrated to this country, to WashU, and where he started his career in atherosclerosis. He started doing imaging and pathobiology of atherosclerosis at that time. About 20 years ago, he moved to University of Kentucky, and where he's continued to do spectacular work in atherosclerosis. One of the things he's done is to give us all in the scientific community an animal model of aortic aneurysm that's quite good. He'll be talking about that animal model of aortic aneurysm today. Dr. Daugherty is the Linda and Jack Gill Professor of Preventive Cardiology, and he directs the Saha Cardiovascular Research Center. He's the chair of the Department of Physiology and the Senior Associate Dean for Research in the College of Medicine there at UK, and Associate Vice President for Research. Also, he's the editor of ATVB, the American Heart Association journal, Arteriosclerosis, Thrombosis, and Vascular Biology. Alan, come on up here and let's hear what you have to say today. Thank you for coming.
Thank you. Okay, hopefully, is this switch on? You're going to just John, thanks a lot. I got to admit, I had an unusual level of hospitality last night. It was a great function around at his house, and got to see a lot of old friends like Henry here that I've known for many, many decades. Well, we're not that old. 2 decades? Okay, 2. Also, I did get to, as John mentioned, I'm the Linda and Jack Gill chair, and some of you might know that Linda and Jack actually are in Houston. So I actually got to see them last night. Linda, in case you don't know, it's stuck between these 2 bald guys. Linda and Jack have done a huge amount for the University of Kentucky in terms of their donations, and more importantly, actually, their activity. This is actually Jim Muller, who recruited me to the University of Kentucky 20 years ago. He was a major instrument in getting Jack and Linda together. And there's Jack between my wife and Jim's wife. As I say, unfortunately, I was hoping they would be here today just to show the pictures. They come in late. I'll make sure I made a big deal out of Linda and Jack. Actually, Linda also was a huge player in Horacialgia, which is an organization they're actually traveling to tonight that helps very underserved students get a great start in life with these scholarships. And I just wanted to make the pitch that they were also sort of helpful, very helpful in recruiting this guy, Mike Carf, to the University of Kentucky. And Mike came from UCLA. He revamped a podunk little medical center to a place that's thriving incredibly today. Not quite as big as this place, but it has gone up to a 1,000-bed hospital these days, and it's a very thriving clinical enterprise. And again, I would really credit Linda and Jack for having a huge input in terms of enthusiasm, in terms of what they've injected into our organization. So I feel pretty much carrying coals to Newcastle here, coming to this place to talk about aneurysms. But as John mentioned, I'm just going to briefly mention this abdominal model that just happened to fall into our lap a few years ago. And actually, I'm mostly going to talk about thoracic aneurysms. And I'm mostly going to talk about it from my angiotensin perspective, of which I sort of going to circle back to this about, is this molecule, which is not new, the original publication that demonstrated the presence of the renin-angiotensin system was published in 1898. So this system is not new. This molecule has been known for a long, long time. But there's an amazing amount of new biology that's coming up with this molecule that's been known for this length of time. And in terms of whether it's relevant to human, again, I'll circle back to this right at the end. But there are, I'm not going to go through these in detail, but there are genetic bases for thinking that angiotensin responsiveness may be a major factor in abdominal aneurysms. The pharmacology is actually a little bit mixed. There has been a study in Canada that demonstrated that people on entry to hospital, if they hadn't a rupture or not had a rupture, there was a much greater presence of ACE on those ACE inhibition given to those individuals who weren't ruptured. There's also a retrospective analysis done by people in Imperial College in London, where they looked at expansion rate on individuals that were taking ARBs, showing that there was a positive benefit there. But a bit of a fly in the ointment is a group also at Imperial in London that also did a retrospective analysis looking at ACE inhibition, stating that actually the ACE inhibition patients had an increase in aneurysm expansion. And really what we're kind of lacking is the ongoing and completed now trials showing ACE inhibition and ARB inhibition. To my knowledge, again, I'm a very sophisticated audience I'm talking to here. There may be more trials in this. The two that I'm aware of is the TEDdy trial that actually is based in your old place in Stanford partly, and in Australia and in Holland. That is recruiting patients taking ARBs. And then the ACE inhibitor trial, the one that is most well known is the Adventitia trial, which is run by my editorial colleague, Janet Powell in London. And basically, this came to this was done because obviously their retrospective analysis was potentially had sort of devastating consequences, because if ACE inhibition was really promoting the expansion of aneurysms, that really needs to be defined very carefully. This trial was not very highly powered. There's only about 250 people in this, and there's 3 arms. But I guess the main message from this particular trial is that retrospective analysis of ACE inhibition causing expansion didn't come out in a prospective trial. It also, quite frankly, didn't show a great benefit of ACE inhibition in this prospective trial, but again, there might be some reasons we can talk about that tailing back to the end of the talk. So from my own perspective, I moved to the University of Kentucky because we had this great lipoprotein group there that seemed to be great for me to interact with for atherosclerosis. But as soon as I got there, there was a RFA from NIH. The basic drift of the RFA was that blood pressure and atherosclerosis are linked, but the sort of black box in the middle was very unclear. So they were calling for applications on hypertension-induced atherosclerosis. And I'd got to know Lisa Cassis, who's my long-term colleague and friend at University of Kentucky, who has been working on angiotensin for many, many years. And it just seemed to be a natural alliance for us to infuse angiotensin into hypercholesterolemic animals to see if that blood pressure augmented athero. I'm actually not going to go down that road, although that's another part of my lab, which we do look at angiotensin-induced atherosclerosis, which is a very profound stimulus independent of blood pressure. But what started happening is that these very short infusions here were causing into these hypercholesterolemic animals, of which they were both LDL and ApoE, again because our endpoint was atherosclerosis, was that we were starting getting these big bulges in the abdominal segment. And the reason that this came of interest to us is because when I was at WashU in the mid-90s, I was working on lipoprotein modification. And the group of vascular surgeons at WashU, especially Rob Thompson, were supplying me with a lot of endarterectomies and at that point a lot of open aneurysms. And Rob and I had these long chats about how this is a very prevalent disease. And the bottom line is that in those days, the amount of research dollars and research papers on abdominal aneurysms was really minimal. And one of the deficits of the field was a lack of an animal model. So these bulges came up, just seemed to have a one-on-one sort of link to us to think of this as being a very nice model of abdominal aneurysms. One of the quirks on this that I was just going to quickly mention is, as I say, this was all hypercholesterolemic-based animals, and that was because that wasn't the aneurysm wasn't the aim of the study. We have subsequently gone through finding out why is it that hypercholesterolemia seems to be a factor that augments the presence. So you can still get this disease in normal cholesterolemic animals, but the hypercholesterolemia markedly increases the incidence. We have just published a very extensive study that Jing Liu, who's a graduate student who's come to the University of Iowa, looking at manipulations. And just broadly, the bottom line is that if you have an augmentation of ApoB-containing lipoproteins, you don't need a huge augmentation, but some form of augmentation, you do get an increased presence of these aneurysms. But a lot of people have come to me and said, that's a pain in the butt, because if you're looking at your gene of interest, that means you've got to put that gene of interest into a hypercholesterolemic background. And just to augment this is a short editorial that I wrote with Ira Tabus and Dan Rader, in which we point out the obvious, that if you want to do these studies in a hypercholesterolemic background, it takes a long, long time to develop these animals and actually do the study. But we did this editorial in combination with some studies in which we were talking about a PSK9 gain of function, which now allows you basically to do this experiment almost instantaneously. Because basically, you're probably very familiar, PSK9 has exploded on the scene recently in terms of a therapy, but on the opposite side of it, it's also become a very good experimental tool. So PSK9 is basically a feature that couples the LDL receptor, and then if it's linked to the LDL receptor, rather than the LDL receptor being a recycling protein that comes to the cell surface every 15 minutes, it instead gets degraded. And basically, a high PSK9 leads to an LDL receptor deficient state. Hence the reason that John and Mary were just talking about this, obviously the great interest now in PSK9 antagonism, especially with the recent trial that was just published last week from the ACC. But what we did was we built on two publications, one from ATVB and one from a lesser journal, in which we did a gain of function mutation, where this was done by Jakob Benson when he was still at Aarhus in Denmark. What Jakob did is he created an AAV which overexpressed this gain of function PSK9 mutant and demonstrated he could acutely make animals hypercholesterolemic. And in fact, just to advertise anyone who wants to do this, this is now a very easy process because interacting with Dan Rader at the University of Pennsylvania, we've made this PSK9 AAV that overexpresses, and it is a very trivial task now to do an IP injection of a virus that costs about $3, and within a week, you'll have sufficient expression to drive a hypercholesterolemic state. So that difference between spending years and years driving things, it makes it very, very different. And again, we've interacted with many people on this now, and we're very happy if anyone wants to engage with us. My colleague Hong Lu, who is the first author on this, has been distributing this AAV many places. The second point, and actually the final point about the abdominal, again, I'm probably preaching to people who preaching to the choir here, is that a lot of papers that come to us at the journal and other forums talk about what is the mechanism in using tissue acquisition and then tissue characterization as defining the mechanism of this disease. So we published this some years ago, demonstrating that there was characteristics of the aneurysm that had some similarity to some aspects of human. I guess this is the issue that I wanted to just briefly mention, though. So this is a paper done by my very long-term colleague, Deborah Terry, in which she sectioned these angiotensin-induced abdominal aneurysms throughout. And the bottom line is you can see that it depends where you take it as to what conclusion you're going to come to. And again, those of you who've done humans, in my experience, which was quite extensive at one point where I did a lot of human sections and went through them, is that characterizing what's an aneurysm is a little tough because it didn't take too much for you to go a different area to have a very different aspect of a very different pathology depending on where you were. So actually, let's say that I knew John was going to mention the abdominal segment, so I mentioned that very briefly, but actually I'm mostly going to talk about our work where we've moved up into the thorax here. And again, probably carrying coals to Newcastle, but you probably know that the first person that was ever documented to die of a ruptured aneurysm was from my mother country, King George here, when he's on the throne. He's on the throne in two ways. He's on the King of England, and he's also sitting on the throne. Good to see a Scottish guy understands that.
Yeah,
okay. Anyway, other than I like saying that story, I guess the main reason I make that point is that if King George was alive today and went to get some therapy in terms of a medical therapy, we'd be about as knowledgeable now as we were when King George popped his clogs as well. So, it's an amazing how ignorant we are about some of the basic mechanisms of these diseases. And, these diseases are really let's say, they've been very central to me in the last few years. It's a really interesting spectrum of diseases. And of course, even with just within that thorax, there's a whole spectrum in terms of the way that these different diseases can occur at different points. So the heterogeneity of this aorta, even within this restricted segment, obviously leads to a huge variation in the manifestation of the disease. So we kind of got into this partly. Does anyone know where Abraham Lincoln was born? Indiana. Nope.
Oh, Kentucky.
Yes.
Sorry.
he moved to Indiana. So part of the reason I say this is because some of you might know that there's a lot of speculation that Lincoln had Marfans. And in fact, Marfans is a defect of the fibrillin-1 gene. And part of the reason I bring up this local link is that the linkage between fibrillin-1 and Marfans was actually made in Kentucky because we have a very high prevalence in the eastern part of the state. So Reed Peretz and Hal Dietz came down and used a patient grouping in Kentucky to really make that discovery. So Marfans is a very common thought of us in our clinic. And it sort of really hit us many years ago when Hal Dietz's group came out with this publication. So Hal's at Hopkins, and he'd made this haploinsufficient animal to mimic characteristics of Marfans, in which you can see that there's an increase in the ascending aneurysm diameter. These are mice that are about a year old that have this haploinsufficient animal model. So Hal very much was focused on the TGF-beta aspect of this, which I'll mention briefly. But the reason it really got our attention is that this is actually the title of the paper. Sorry, it's not the title of the paper, but the paper showed that losartan, which is a classic AT1 receptor antagonist, reduced the, almost ablated actually, the development of the disease in this particular model. And obviously, since we were very angiotensin-centric, this really caught our attention. And I just wanted to mention, actually, for some practical reasons, again, if anyone's getting into this, about some techniques in terms of looking at this in animal models. Because one thing that's become very central is the measurement of this in animals. And it's become very central using these great machines that have come up. So the Visual Sonics, a company in Canada, developed initially the 660, and it's now gone through a series of evolutions, gone from analog probes to digital probes. And the reason that I'm sort of getting into this is this was great when we were looking at the abdominal to get the 660, but measuring aortic diameter up in the thorax of a mouse has got a lot more complications and really requires some of these digital machines, which unfortunately are astronomically expensive. But part of the reason I'm trying to accentuate this is if you contrast the analog to the digital images, you can see, hopefully the light's not too much to blank out these images, but you can see, and if I just blow this up a little bit, the definition with these things like the 2100 for the wall thickness is just so much sharper and better. Keep going. And the other thing I just wanted to accentuate in terms of the quality of data that you can get from this is that we have a lot of discussion about how you measure it and where you should measure it. In fact, Mary Shepherd, my colleague here, is working with a group that we've got and sort of trying to develop some criteria for coming up with these animals in terms of getting some realistic measurements of what was considered to be a dilation. And the reason I bring this up is, so for example, in this original publication, which used the Vivo 660, you might see this excursion up here is about 0.13 millimeters between systole and diastole. And in fact, this is a sort of a hallmark paper looking at the effect of TGF-beta neutralization. And in fact, the difference there is about 0.15 millimeters. And so in terms of being able to define that this is actually an effect, we've really realized you've got to be extremely careful about how you do this measurement. And it does kind of depend on context, because some data generated by Murray and Jeff Chen, who's an MD PhD in my lab, sort of demonstrates that this, for example, the systole diastole can vary very much in wild type versus, in this case, some fibrillin-1 hypomorphic animals who have a very rapidly expanded ascending aorta. Okay, so that's a little bit of technical aspect that in terms of we've found to be extremely important to get data that we think is reliable. But actually, there is a lot of mouse models of ascending. Unlike here, where we're a bit restricted, up here, there's a tremendous number of mouse models. And just like humans, they have some regional specificity to them. You might gather, obviously, since I'm an angiotensin-centric guy, that angiotensin has sort of been central to what we've been doing. And the basis of this partly was because of this study that got into it. The other was that Hal's group also did this retrospective analysis where he took 18 kids, and then these kids were on standard of care of all on atenolol. Then at some point, probably not for great reasons, they were switched to an angiotensin receptor blocker, which in 17 of them was losartan, and one of them was irbesartan. And then the inflection point that you can see there is where the kids went on to the ARB, showing that in this group there was a tremendous reduction in rate of expansion. And in fact, between Hal's mouse study and this retrospective analysis on a small group of kids, that spawned a whole group of trials, which I'm putting this more for effect than anything needing detail. But there is many trials in 9 different countries now looking at the effect of angiotensin receptor blockade in thoracic aneurysm, primarily in Marfans, but also in BAV patients. So I guess I'm going to go through sort of about half an hour of where we're trying to go in terms of, is that angiotensin response, how is it affecting the disease, and has it got any reality to human disease? So first off, the same model I mentioned before, just infusing angiotensin, but whereas we focused in the early days all down in the abdominal region, we kind of realized that exactly the same model causes these very profound pathologies up in the thorax as well. And although we initially started this data all in hypercholesterolemic animals, certainly for the thoracic disease, that is irrelevant. We've now switched to just normal lipidemic animals, which makes life a whole lot easier, and also probably a lot more relevant because clearly that's hypercholesterolemic state is not really known to be related to the presence of thoracic aneurysm. So when you infuse angiotensin into animals, you get some really bizarre pathology that's very located just in the thoracic region. So you can see this example on the left where basically at the branch of the subclavian and distal to that there is, it's very ordinary, but up through especially the ascending portion, you can see these really weird segments that you get. So can be a very, very profound pathology that frankly we missed for quite a long time. So we've gone back and my lab does a lot of sequential pathology that we call, where we think it's very informative to look at the different stages of the disease and find out whether the pathology differs at those stages. And again, one thing that we missed early on was that if you just do a very short interval of angiotensin infusion, then bring these tissues out, you can see this not very subtle pathology that is present. This is a very determinable red cell count accumulation. The thing that's really interesting about this, or at least I do, is when you do the cross sections, because this is this beautiful Movats immunostaining with the lumen on the upper side, the Adventitia on the outer side. And this happens all the time, where when you get this blood, it only accumulates in the outer 2 marginal layers. So it's somehow getting there from routes that we cannot define. We've had several people just sitting, going like this, trying to find out whether there's an intimal flap, for example, that was the entry point. We have not been able to find an entry point in the lumen. Even if we did, frankly, it's a little confusing as to why that would just accumulate on the outside laminal layers. Why would it stop there? Also, getting it from the adventitial side is a little confusing as well, because mice do not have medial vas vasorum, or at least I've never seen them defined as having a medial vas vasorum. So getting it through the outer laminar layers is very confusing, but might be very important to how we're operating here. So if you keep on infusing these animals, again, if you look at the contrast between a normal and a infused one, you can see that at the branch of the subclavian here, everything after that is pretty much dimensionally the same. It's a little more opaque because you get some adventitial thickening and some medial thickening. But you can see that in the ascending portion through the root, there's this not very subtle expansion rate. And again, if you do a cross-section of this, it's got this pathology where on the inner layers, things are relatively normal. Then as you get further and further out, you get more and more bizarre histology. So one of the things that's come up about this aspect is people say, well, mice are kind of different because they're so small. And in fact, that is definitely the case, because these classic studies that Seymour Glagoff did years and years ago, where he looked at the structure of the aorta from elephants to mice, and just looking at laminar layers and different aspects of it. Glagoff pointed out that up here in the human laminar units, they're usually in the range of about 40 to 50. And it's usually only about the outer 20 that have medial vas vasorum going into them. Whereas clearly mice up here only have about, actually usually about 8 to 9 laminar units, and I don't know if anyone's detected any medial vas vasorum in that segment. So there are differences there, but we've actually done quite a few human thoracic aneurysms. Mostly our collaborator is John Elefrades, who's got this very large tissue bank up in Yale of about 1,000 patients, mostly BAV repairs. And certainly when we've sectioned a lot of these samples that John sent us, we also get this sort of gradient in terms of pathology, where it seems like the luminal aspect usually seems in these tissues to be much better shape than the adventitial aspect. So part of our basic biology here is saying, we get this very reproducible disease. What the hell is it caused by? And can we really define this by first off defining the receptor that's causing this? So in terms of angiotensin receptors in mice, they are a little different from us. So the AT2 receptor is the same, and AT2 receptors are a little bit of an enigma because they're very highly expressed during embryonic development and basically switch off, and the functional significance is a little unclear. We have got the ability through Tadeu Magami's studies to take out the AT2 receptor, and we have, through Dr. Magami's collaboration, looked at the role of AT2 receptor antagonism in this model, and we cannot find any difference in AT2 deficiency versus non-AT2 wild type. There is a drug, PD123319, which is an antagonist of the AT2 receptor, and again, we've used this in this particular model. We have not been able to show any effect of AT2 antagonism. In us, we only have one AT1 receptor. In mice, there's actually a chromosomal duplication of this, but both of those receptors are indiscriminately inhibited by sartans. And Tom Kaufman, who's has been head of nephrology at Duke, did take out the AT1B receptor and wasn't able to show an effect. So it's fairly clearly that the pathology we see is an AT1 effect. I should just mention, actually, again, my colleague Mary Shepard, who's sitting here in front of me, she is getting into the AT2 receptor in a slightly different way, because some of you might know that Hal Dietz's group had a very high-profile paper in 2011 in which the discussion was that the AT2 receptor agonism would be a new therapy for individuals that are afflicted with thoracic disease. And in fact, we are undergoing some basic trials which are complementing some, what we gather, clinical trials going on the role of AT2 agonism in this particular study, using this compound C21, which was developed by a group in Germany and is now being developed by a group in Denmark. So back to our work in terms of the AT1 receptor. So you delete the AT1 receptor during the infusion. You can see the expansion you get there, which is totally ablated in the absence of AT1A. We have been trying to think, what does AT1A do and why is it localized to that area? So some of the earlier studies we did where we just took out the aorta and we formed rings, and then we looked at whether those aortas contracted in different regions. The basis being, if AT1 is involved, is it because the AT1A causes great contraction up in here, or greater responsiveness manifested by contraction? So we did these studies where we took out the aorta, took out small rings. We did this contractile profile of potassium, 5-HT, and angiotensin. And in fact, this was a little surprising, although there's been many studies now that have been very consistent in this, in which if you take out the ascending aorta where our pathology was, and we thought this is where the AT1 receptor was really going to be, and then you look at this profile of potassium, 5-HT, and then angiotensin, in fact, there is no measurable contraction. And several people now have demonstrated very similar data. And in fact, again, in this aortic setup, the only place that you can get any contractility is down here in the abdominal segment, which is a totally disease-free area in all the models that have come up. So even though the AT1A receptors clearly the receptor that's involved in it, certainly in terms of responsiveness based on contractility, that didn't quite work out as giving us any insight. Another study that we did was by Phil Owens, who's now just started up his own lab in cardiology at the University of Cincinnati. Phil did a study where in vivo, he said, okay, we'll infuse a bunch of angiotensin in vivo, we'll take out these aortas and see whether we can see any regional responses that give us some insight as to why the disease should be located in that area. So this is an experiment that was done in a very anal retentive way that Phil tends to do, where he took out the aorta and sectioned them very carefully and did multiple measurements. And the first aspect of it is I've just taken selected data where he took the aortas from saline-infused animals and angiotensin-infused animals and then looked at the thickness. And the bottom line is the thickness is pretty equivalent right throughout the aorta. There's no regional discrimination that goes, wow, you know, angiotensin must be really hitting that part of the aorta and causing a very different response, at least based on this criteria. However, Phil also looked at the nature of the tissue. Some of you might know there's sort of a bit of a, I don't know about dispute, but there's very disparate data about how angiotensin affects smooth muscle as to whether it's causing a hypertrophic effect or a hyperplastic effect. And in fact, the answer is yes, because it depends where you are. Because certainly based on Phil's data, when we looked at what caused that thickening in most of the aorta, it seemed like hypertrophy. But in the ascending, actually, even though the dimensional changes were kind of equivalent, it seemed like it was a very different mechanism based on the number of nuclei, which doesn't in itself mean hyperplasia because you can get multiple nuclei for many different reasons. But it just demonstrated there was something weird about this particular segment. So what this meant was a many-year push to say, okay, that's not really giving us great insight as to why we get something right here. But maybe we should start taking the receptor out in different cell types and then see whether we can see different responsiveness by deleting those cell types. So we had to, unfortunately, develop our own mice, which is a long, expensive, tedious process where we flox the AT1A receptor. Turns out that Tom Kaufman was doing exactly the same experiments at the time and developed pretty much the same animals, which in some ways has been a waste of resources, but actually we've both got incredibly similar data, so it's sort of been complementary and helpful because it's been a little surprising. What we did was the same as the original Oliver Smitty's model, where we put the flox sites either side of exon 3, which is where the entire protein is translated, so this is an entire deficiency. And actually, the only thing I'll accentuate is that we did get a very efficient deletion. Those of you who know this field know there's a little dispute about the antibody world in terms of knowing whether you really have an efficient protein deletion. And in our case, we cannot get any of the antibodies for the AT1A receptor to work. They're present in about 1,000 publications from different sources, but we can't get any of them to work. So we weren't able to determine at the protein level that we had efficient excision, but we were able to do it at both the RNA and DNA level. So these, actually in the early days, we were still doing hypercholesterolemic animals. So this is a very tedious breeding process of making these mice, which is about a year, year and a half, and then breeding them, which is about another 2 years, and then developing these animals in sufficient numbers to do the study. And just basically what we did was to take out the AT1 receptor in both the endothelium and the smooth muscle. So we were really expecting to take out the AT1A in smooth muscle, and this was like a no-brainer. We would get this huge effect in reducing the extent of aneurysms. And in fact, this is an example of the data that we got, where you can see the mean data to the left, and the individual data to the left and the mean to the right, looking at whether on the left they had the AT1 receptor in smooth muscle, and to the right had it deleted. And after all these time, money, and effort, this was an incredibly disappointing experiment to show that what we thought was an obvious slam dunk would not be obvious at all. So we cannot find any effect of taking out the AT1A receptor on this type of pathology. And again, I mentioned the Duke group also was totally unable to find any change in their pathology taking out the AT1A receptor. So despite its inherent obviousness, it didn't quite work out that way. But one of the areas in the thoracic world is that there is that outside area that was kind of ignored for a long time, but clearly there's a lot of interesting stuff out in the Adventitia. And in fact, this kind of came a little bit to level some of the colleagues that just down the road from here, both Alan and actually Ron Tilton was one of my former colleagues in St. Louis. Really sort of one of the groups that have focused on the fact that the adventitial fibroblast may actually be the provoking factor in the disease that they see down in Galveston. In fact, I didn't know who would be here, but just as a small world on this, that Telhar is actually an undergraduate student in my lab, has just finished his PhD as part of the MD PhD program in Galveston and was working on fibroblasts and ascending aneurysms. So shows there's lots of little family connections here in science. But anyway, we also then started this, again, quite long study in which we took out the AT1A receptor in fibroblasts. I've put the quotation marks of specific here, because those of you who do this work probably know that that's pretty much an oxymoron, a specific Cree. These are clearly not specific, but certainly when we started this work, the ability to delete something specifically in fibroblasts was limited to this particular one promoter that was developed at Vanderbilt. It has both two issues to it. One is the potential for lack of specificity, and the other is that fibroblasts are actually an incredibly heterogeneous cell type, and there's great variation in it. So we're not actually clear that this S100 is taking out the gene in every fibroblast. Having said that, this study that was done by Aruna when she was in the lab before she's now a postdoc with Christy Redhorse in Stanford, Aruna infused angiotensin into these mice that had the AT1 receptor deleted, or wild type or deleted in the fibroblast. And what you can see in the ascending portion is that in the bluish graphs that show the angiotensin caused the expansion in terms of the thickness, when you deleted the AT1 receptor in the fibroblast, that particular region had almost a complete ablation of that thickness expansion, which was, again, a little surprising to us, but this has been done quite a few times now. And it was a specific effect to the ascending portion. Taking it out and the descending didn't have that effect. So what seems kind of strange is, so we always went into this thinking angiotensin would be hitting the smooth muscle, causing this very profound effect in pathology. But actually at the moment, it seems like that angiotensin is hitting something, fibroblast on the adventitia, and then causing some profound effect in the smooth muscle, which is not what we expected going into it. But I've got another story that seems to really accentuate that, because we clearly show that you take out the AT1 receptor in fibroblasts, you have a difference in pathology. But then we've been working on this other protein in smooth muscle that has this very profound effect on the disease. So this, to me, is one of the most interesting proteins in biology. It's called the low-density lipoprotein receptor-related protein, or LRP1, much easier, rolls off the tongue better. This is a tandem repeat of the LDL receptor that was pulled out many years ago by two groups independently. One in Heidelberg, Joachim Herz, who originally gave it this name, and then a group in Urhaus in Denmark who found it in a different way and called it the alpha-2 macroglobulin receptor. The bottom line is, part of the reason this is so interesting is that there's select ligands on this particular protein, but actually it's not that selective. It's kind of considered in the scavenge receptor class. Basically, if you lack this protein, which no human does, but if you take cells out that lack this protein, it affects the proteolytic environment of every cell in the body. And so, fortunately, Dudley Strickland, who's a close colleague and friend up in the University of Maryland, who actually published this article in ATVB, gave a talk a couple of years ago at AHA. And, oh, sorry, just to mention, by the way, this is your colleagues down here, actually, in Texas, have just been involved in a study that has illustrated that LRP1 polymorphism can have an effect on ascending aneurysms. And in fact, I do know there's other groups that have very, very nice data showing that a functional domain of LRP has a very profound effect in the presence of a thoracic aneurysm. So there is a definite human correlate here. So when I happened to bump into Dudley, he'd developed these LRP deficient animals in a smooth muscle specific fashion, and was good enough to provide us with all these animals, send them to Kentucky, for us to do our standard of experiment where we'd infuse angiotensin and see whether they had a difference in pathology. So this is looking at something just in the smooth muscle now, even though the angiotensin effect doesn't seem to affect the smooth muscle. Those of you who might know this field know that my colleagues just up the road in Cincinnati have published on these animals. And if these animals are quite old, they actually do have ascending aneurysms spontaneously. So it was important for us to know at the age that we did our animals, which were very young, they actually had a very normal type of thoracic aorta when we looked at them. But then we started doing multiple studies on these. One was that we have a 7T where one of our friends, Brandon Formwald, helped us out do these full body reconstructions of the aorta. And there's a few weird things that came up with these animals. One I'm happy to talk about, some other people, is this down here, where actually there was a huge presence of mesenteric aneurysms that occurred in these animals. But it was actually up in the thorax that I'm just going to focus here, because if you look at the raw images, you can hopefully see, if the light's not blanking these out, is that the image on the left, which is a normal animal, has a very different arch structure from the animal that's on the right, which not only has issues in the ascending, but has tortuosity occurring. And in fact, if you do the reconstructions of these, you can see that the pathology that occurs up in just that limited segment is quite profound. And in fact, when you do the dimensional changes of this was done by a student, a Sarnoff fellow that was in my lab for a year, Frank Davis. Frank did these beautiful studies where, if you take out the aorta of wild-type animals and look at the thoracic area, it's fully unsensational. If you infuse angiotensin, you get this very profound expansion. An LRP1 smooth muscle deficient animal infused with saline will look pretty normal. But then if you do the combination of the two, there's a huge augmentation effect. And in fact, in terms of dimensional changes, if you look at the saline infused between wild type and knockout and look at the same animals, this is a 0.4 millimeter change, which, to try and put that in the context of the relative change that I mentioned going through these instruments, this context is well within the accuracy of this type of measurement, and it's very, very large. So we got this situation where you seem to have the Adventitia being stimulated by angiotensin affecting something about the smooth muscle. But another aspect of this is that that gradient that we see, and is it just an aspect of because the stimulus is from the adventitial side, or is there something inherently different about the smooth muscle cell populations that populate this area? So I'm now, Mark Majewski is at Seattle Children's, has been a long-term friend and colleague of ours, wrote this classic ATVB review, which is oft quoted, demonstrating that the aorta is actually populated by smooth muscle of many different embryonic origins and potentially has different functional consequences. And in fact, if you just blow up that portion that Mark did there, you can see that this is a classic interpretation of the thoracic aorta, where right down in the root, those smooth muscle cells are populated by ones that originating from the second heart field, whereas the continuum of the rest of the area through to the subclavian is populated by smooth muscles of the cardiac neural crest. And in fact, if you look at the distribution of the cell type origin, this is a picture of angiotensin-infused aneurysms in this region. And this is a paper at the USC group that came out a long time ago now, and it's kind of a standard slide that everyone has to give when you give these talks about ascending aneurysms. You can see that the distribution of cardiac neural crest, defined by the blueness of the beta-galactosidase staining, matches incredibly well with the appearance of the ascending aneurysm. And so there's been a lot of thought that there's something about the cardiac neural crest cells that, in terms of its responsiveness, that is responsible for this incredible co-localization of disease. But one thing that Mark noticed many years ago in this paper that he had in 2008 is that when he did those studies where he marked cells, the cardiac neural crest, they looked the same image that you just saw there. But when he did a cross-section, again, I don't know whether with the lights you can see this, but there's a blueness, but it's not consistent throughout the aorta. And he assumed that was because maybe there was a bleaching or just a lack of sensitivity and didn't really think at the time much about the fact that the blueness was only restricted to the inner layers. So in fact, we've gone back and redone these studies in collaboration with Mark, with a MD PhD Japanese cardiologist that's a fellow in my lab, Hasashi Sawada. What Hasashi's done is he's rebred these animals. So we're looking at where smooth muscle cells populate in this specific circumscribed region. And basically he's taken these Rosa 26 lac-Z mice, bred them with this Wnt1, which is a classic marker for the cardiac neural crest. And this is a picture that Hasashi's got, identical to what was published in 2000, right down to where it goes through, where it goes down the DA, the ductus there. You can see it's positive. But the second heart field, amazingly, we've used the MEF2C in terms of the Korean, and there's some debate about this, and love to talk about this. Again, in terms of the wild type animals, we don't get any positive background. But as soon as we go to the MEF2Cs, we get a very different distribution from what was brought up in that classic paper by Mark. And this fact that the second heart field would just be located right down in the root, and that transitional area between the root and the sinotubular area would be part of the positional location for aneurysms. We're not quite sure that's true, because again, I don't know how obvious this is to you, but at the anominate now, basically the second heart field goes right up the ascending aorta. And it's the same in males and females, and again, those of you who know this field, this is actually quite different from some earlier publications. One of the issues is maybe it was an early age discrimination, and then as you got later, things changed. But in fact, we've gone to quite mature animals now, and it's amazing that this distribution stays consistent throughout that whole period of time. And in fact, when Hasashi is doing these studies, it's very clear that if you look at the second heart field, they are on the inside surface, just like Mark showed a long time ago. But in fact, the smooth muscle on the outside of the entire ascending aorta, in our experience, seem to be populated with a very different population. So unlike the model that Mark originally published in 2007, actually this is still a little superficial because we've got through a lot of nuances, but I think there's quite a big difference in terms of the way that this area is populated. And I guess it doesn't probably surprise anyone that we're now taking LRP1 out in these very specific populations to see whether we can find a regionality to the reason that these aneurysms are occurring in this particular area. So let's say I'll just quickly sort of bring back to some of the human issues that I think are discussed, because my lab plays around with mice and gets me grants, but I would like to think we do things a bit more altruistic than that are actually purposeful. And so in terms of whether angiotensin is involved in human aneurysms, obviously my world is pivotal to this in terms of, but I would totally hold out that there's obviously ambiguity about this, but it's a case of why is it ambiguous. So actually in the animal world, it's totally unambiguous because I mentioned there's many models of ascending aneurysms that actually have amazingly consistent pathology. And in all these studies that have been done, the Mount Sinai studies and the hypomorphic studies, the Hopkins studies, and actually in the lower part here, the studies done on this campus, those studies that have shown you get ascending dilations, every one of them is responsive to losartan. So there's absolute uniformity about the data in animals. It's amazing uniformity, actually. Of course, all these human trials that I mentioned, there's now about 3 of them reported out, of which the first one was the COMPARE trial, came out of Holland, and I think gave people a lot of hope that this would be positive because of the decreased dilation in that particular study. But I think this is probably the one that's hit people most, is that the trial that was started off based out of Boston Children's, run by Lacroix, that was reported out at the AHA a couple of years ago, looked at the contrast of atenolol versus losartan in a pediatric population. And the bottom line is that to the great disappointment of the community, that there was no superiority, in fact maybe a slight inferiority of the losartan administration versus atenolol. There's been a lot of discussion about the way that atenolol and losartan was given in these particular studies, but that's going to be one of the debates that's that's needed to have, because one of the things that I would contend is that losartan is a not very good drug. I mean, it's the daddy of this class, but it's incredibly short half-life. And maybe just as important as that, this concept of surmountable, insurmountable antagonism. So losartan is a surmountable antagonist. So it can be easily overcome by local excesses of angiotensin II. And in fact, we'd be much better served by thinking about drugs that have a much longer half-life in combination with the thought about the surmountable antagonism. Again, I don't know whether I'm preaching to the choir here, because I was at the Japanese Circulation Society last week, and one of my former trainees is a pediatric cardiologist in Saga in the south part of Japan, and he has a lot of Marfan patients in that area. And during discussions that we had at dinner, Katsuya was telling me that he gives all his patients candesartan because losartan is such a poor drug to give because of its short half-life. I don't know that mindset is very prevalent, but I think it's incredibly important in terms of, I've got a feeling that we might be coming to some negative conclusions, but maybe not for the right reasons. So, I won't go through this, but the last part here is basically, we really have to think about selecting the right drug if we're really going to test this hypothesis. And unfortunately, most of these trials are not probably using the optimal drug. The only one that I'm aware of is the British trial, which is mired in red tape. So proud of my mother country. But that is using irbesartan, and maybe once that actually reports out, that might be a really interesting trial to contrast what might be very variable results in some of the other trials. So I think I've blabbed on for long enough. I just wanted to get over that Lisa Cassis has been my longtime friend and colleague down at Kentucky, that she and I have worked on this together for many years. Mark is now my co-grant holder in terms of doing this work. Frank, those of you, I don't know if any of you have heard of the Sarnoff Foundation. I'm now into the scientific committee for this group. This is a great organization that takes med students out of their current place and places them in a different institute for a year. I was fortunate enough to get Frank out of the University of Michigan to work with me for a year. He's now a second-year vascular surgery resident at Michigan. And Phil Owens, who, that's a rather youthful picture of him, who is now a faculty at Cincinnati. I'm not going to mention all these guys other than the fact that Sashi Sawada up there is his MD PhD from down in southern Japan that's with me for a few years. And Jeff Chen, who I didn't mention too much, but is an MD PhD student, is also doing a tremendous amount of the work. I better just mention that a lot of the name you'll see there is Ruturi, which is this individual is in the center of everything, who is the first person I hired 31 years ago and is still in the lab. I did have to go through the extraordinary retention process of marrying her, but this has now been a very successful interaction. Anyway, I thank you for your attention. Happy to answer any questions.
Thank you, Alan. We have some time for a few questions. Dale Hamilton.
Nope, it's not running.
Why are you getting to Dale?
Nothing.
It's part of the problem in the trials that in addition to the heterogeneity, the vascular smooth muscle, there's genetic heterogeneity. There are a lot of different factors that are causing aortic aneurysms. Tell us a little bit about that.
Well, so these trials that I mentioned, they're actually almost exclusively Marfan trials. So in terms of all these other indications that I mentioned that obviously have a very strong indication with that, there's no clinical trials on that. And it's going to be this assumption that if it's positive for Marfan's, if it is positive, it'll be positive for the other groups, and counterwise. But those clinical trials are not going to be very easy to do. It's like getting the proof is going to be tough. Yeah.
This is a question about the role of nitric oxide in the development of these aneurysms.
Uh-huh.
We use an angiotensin II model for heart failure, but we add L-nane, inhibiting NO formation. And we do not see changes in the same mouse model, the C57 mouse model. We do not see changes using the Vivo 2100 in the aortic root there, using the combination of Ang2 plus L-nane. Have you looked to see if inhibition of nitric oxide formation would prevent the formation of these thoracic aneurysms?
No, we have not, but there are a few other groups that are very actively looking at that, because that might be part of the contrast you've got there from our data in terms of not getting expansion. So there is a group up in Vancouver that's been very actively looking into the role of L-node. Oh, behind you.
All right. Of course. Well, first of all, thank you very much. So I'm on the surgical end of this, and I'm sure, speaking for the surgeons, we're all sitting here thinking, is this the same disease that we really are dealing with on a daily basis? I mean, I think the Marfan's patients are a relatively small subset, albeit very important. So fast forward now into the patients that we deal with, and that's the 60, 70-year-old. And then subdivide them into two different groups. Those are dissections and aneurysms, which we think of as two completely different pathologies. So what are the messages for us in dealing with this? What can we learn from it? Because as we look at your mouse models, and you can create an aneurysm within 4 to 5 days, I'm sure everybody here is thinking, okay, that's a completely different animal, literally and figuratively, from what we're dealing with.
So I think we always have that problem with any animal model. You know, I've dealt with this with atherosclerosis for years. Is there any relevance to this? And it's always kind of hard to know because, for example, in the abdominal area, we tried to take out a lot of human sections in the early days and compare them. But, you know, as you well know, the tissues that you get there are not in great shape. Obviously, that's an extremely advanced disease. Clearly, we are not mimicking every facet of it. So actually, what we're trying to get is it would be really great to have better interactions with people in surgical aspects, looking at tissues and sort of seeing whether we can tweak different variants of these models to see if we can get facets that mimic some features. I think the options of us getting a model that mimics the whole range of dissection aneurysm that you get in humans is probably not likely.
Enjoyed very much your presentation. Obviously, many factors come into play here, but I wanted you to address conceivably the physical factors. Very different in the ascending aorta, in the arch with centrifugal forces, pulsatility, and the laminar flow is different in the arch. So the changes, the differences that you see, in addition to the differences in the type of cells, how much of that can we ascribe to it?
Well, so actually, I guess I should have quite often put a slide up saying, you know, these are all the factors that could relate to that regionality. So you're right, flow is a very obvious thing, and it's a little hard to quantify and measure in a mouse. I mean, I am working with one of our cardiologists at UK who is doing these beautiful images in humans of flow characteristics. But, you know, it's very hard to manipulate is the bottom line, but it clearly has to be taken into account. You know, the extracellular matrix has tremendous regional variances as well that could be a part of the accountability. You know, even the adventitial aspects vary tremendously along the aorta. So, you know, I am focusing down on one area because you have to focus on something, but I would very freely acknowledge there is a tremendous number of issues. And, you know, part of our smooth muscle, maybe it just happens to be in the right position that causes the disease, as opposed to it causing the disease. So, you know, whether it's cause and effect, that's going to be a thing we're going to have to tweak out.
So I noticed on the initial slide that you showed of Mark Majewski's data with the beta-gal mice that the neural crest cells were also involving the proximal trunk to the pulmonary artery. Now, obviously, it's a less sexy area, the right side of the heart, but I was wondering, has that been borne out with the Wnt Cree mice that you have, as well as possibly the MEF2C?
No, it hasn't, but actually, so it's actually deliberately kept on. So very astute of you to look at that, because he felt that was very important. He's redone these images quite a few times to show that aspect of the positive staining. And, you know, we're putting it out there just as a report. But, yeah, I don't know that it's come up with the Wnt1 as well. It seems to be exclusive to the MEF2.
So the reason why I mention that is because one of the standard of care treatments for transposition of the great arteries nowadays is to do a neonatal repair where you switch the pulmonary artery with the aorta. And so the problem then becomes that the neo-aorta is now the former pulmonary trunk. Yeah. And so these patients are subject to dilation and then neo-aortic regurgitation as a result of that dilation. So it would be very curious ultimately whether or not these patients should all be treated with an AT1A antagonist as well.
Yeah, never thought about that. That's very interesting.
Well, let's thank Al for spending time with us today, sharing his information.
Thank you.